Hello friends, today we will discuss about an important topic, that is structural organization of proteins. Proteins have different levels of structural organization. This include primary structure, secondary structure, tertiary and quaternary structure. Primary, secondary, tertiary and quaternary. All of you know the definition of protein. They are polymer of L-alpha amino acids. Okay, and they have 4 levels of structural organization. This includes primary structure, secondary, tertiary structure and quaternary structure. When the amino acids, they are arranged in a linear sequence, for example, this is when amino acids, they are arranged in a linear sequence, it is known as primary structure. Amino acids, they are arranged in a linear sequence, it is known as primary structure. It is made up of amino acids and mainly peptide bonds. Okay. It is a polypeptide chain. When this polypeptide chain is folded upon itself, it is known as secondary structure. It is a spring-like structure with central axis. When folding of polypeptide chain upon itself, it leads to the formation of secondary structure. And when this secondary structure folded to form a 3-dimensional domain, to form a 3-dimensional structure, it is known as tertiary structure. So this is about single polypeptide chain. It can be organized into primary structure, secondary structure and tertiary structure. While quaternary structure means when 2 or more than 2 polypeptide chains or subunits, when they are kept together, when 2 or more than 2 polypeptide chains are kept together, it is known as quaternary structure of protein. Primary structure, secondary structure, tertiary structure and quaternary structure. It is a linear sequence of amino acids linked by peptide linkage. It is known as primary structure. Folding of primary structure upon itself leads to the formation of secondary structure. Further folding of secondary structure to form a 3-dimensional structure. It is a 3-dimensional structure which is a functional protein. It is known as tertiary structure of protein. And when 2 or more polypeptide chains they kept together, it is known as quaternary structure of protein. Now we will discuss one by one with example, that is primary, secondary, tertiary and quaternary structure of protein. So this is an overview. Coming to the primary structure, it depends mainly on number and sequence. The first important word is number of amino acids and second one is the order or sequence of amino acids. Sequence of amino acids. Number, how many amino acids are there? If it is 2, it is known as dipeptide, 3 tripeptide, 4 tetrapeptide and so on. So it depends on number of amino acids and sequence of amino acids. Okay, they are connected by a linkage that is known as covalent bond, which mainly includes peptide bond. Peptide bond. Okay, covalent bond which mainly include peptide bond and occasionally disulfide bond. So these two bonds, they are included in the primary structure of protein or polypeptide chain. Got it? Number of amino acids, sequence of amino acid, mainly stabilized by the covalent linkage, that is peptide and disulfide. Okay. Example is insulin. Example of primary structure is insulin. Okay. Now this peptide bond, it is a covalent bond. It is partially double bond in character. This is CONH. This is NH3 CH CO NH CH CO. This is a peptide chain. This is a functional group. This one is peptide linkage. CONH. This is a peptide linkage. Okay. These amino acids, first, second, third, fourth, they are connected to each other by peptide linkage, and the amino acid, after losing water molecule, they form peptide linkage or amide linkage, and they are known as amino acid residues. First, second, third, fourth, connected by peptide linkages. They leads to the formation of primary structure of protein. Okay. It is read from N terminus, this amino terminal, it is known as N terminus, and this carboxyl terminal, it is known as C terminus. So this is a polypeptide chain. Example of primary structure is insulin. Insulin, it is composed of two chains, that is A chain and B chain. This is A chain and B chain. Structure of insulin, it is sequenced by a scientist known as Frederick Sanger, by the Sanger method. It is the sequenced by Sanger, Frederick Sanger, by the Sanger method. It has two chains, chain A and chain B. Chain A, it starts from glycine, while chain B, it starts from phenylalanine. A, B. Okay. 21 amino acid in A chain, 30 amino acids in B chain. They are stabilized by interchain and intrachain disulfide linkage. Here 7 cysteine of A chain and B chain, and in between 20 and 19 of A chain and B chain. Intrachain disulfide between 6th to 11 disulfide linkage. This is a structure of insulin. It is first isolated in pure form by Banting and Best. Okay, so it is included in a primary structure. It is stabilized by peptide linkage and disulfide linkages. It is also known as branch protein because of disulfide linkage. So this is about the primary structure. It depends on number of amino acids and sequence of amino acids. If sequence of amino acids change, it will lead to the formation of new polypeptide. Then it is stabilized by peptide and occasionally by disulfide linkages. Example, insulin. N terminus, C terminus or amino terminal, carboxyl terminal. This is peptide linkage which is partially double bond in character. This bond, it is known as peptide bond, while this C alpha N, it is known as phi angle. It is known as phi angle. And this C alpha, it is known as psi angle. It is known as psi angle. There is no rotation along this peptide bond, but rotation is allowed on this angle. Okay. So this is about the primary structure of protein or structural organization of protein into primary structure. Okay. Now we'll discuss the secondary structure. It includes alpha helix, beta pleated sheets, turn, bend, and loops. These are the structures which are included in the secondary structure of proteins: alpha helix, beta pleated sheet, turns, bends, and loops. Secondary structure. Of these alpha helix and beta pleated sheet, they are most common conformations. Alpha helix and beta pleated sheets. Okay. Secondary structure, they are formed by the further folding of primary structure. Okay. Coming to the alpha helix. It is a most common stable conformation spiral, right-handed, and stabilized by hydrogen bonds, mainly by the hydrogen bonds, followed by hydrophobic interactions, electrostatic forces, and van der Waals forces. These are the forces which stabilizes the secondary structure: hydrogen bonds, hydrophobic interactions, electrostatic forces, and van der Waals forces. Okay. It is a most common stable conformation, spiral and right-handed helix, alpha helix. Structure is like this. Around a central axis, okay, around a central axis, this is a alpha helix. Each turn, it has 3.6 amino acids, and pitch of each turn, it is 0.54 nanometer, while distance between two amino acid is 0.15 nanometer. This alpha helix, it is stabilized by hydrogen bonds between carbonyl oxygen and amide nitrogen of first amino acid to the fourth amino acid. It is stabilized by intensive hydrogen bonding between carbonyl oxygen and amide nitrogen. of first amino acid to the fourth amino acid. Means this first amino acid, this carboxyl of this first amino acid, it is linked to the nitrogen of the fourth. So these bond, hydrogen bonds, they are main in the stabilization of secondary structure, followed by hydrophobic interactions in between hydrophobic amino acids, then electrostatic forces in between positively and negatively charged groups, and weak van der Waals forces. They help in stabilization of secondary structure. So this is about the alpha helix. It is most common stable conformation, spiral, right-handed, stabilized by intensive hydrogen bonding. And this structure, alpha helix, it is destabilized by proline. It is mainly destabilized by proline and glycine. Also, it is destabilized by when when there are number of negatively charged amino acids which repel each other. For example, glutamic and aspartic acid, or series of positively charged amino acids, that is lysine, arginine, and histidine. They also repel each other. So it is destabilized by proline, glycine, sequence of acidic amino acid, and sequentially arranged basic amino acids, positively charged amino acids. The most common amino acid in alpha helix is methionine. It is methionine. The alpha helix, it is found in myoglobin. It is also found in hemoglobin. Myoglobin and also found in hemoglobin. So this is about alpha helix. Now coming to the beta pleated sheets. These beta pleated sheets, these are extended. These are extended in zigzag manner. These pleated sheets, they are extended in zigzag manner. This is N terminus. This is C terminus. There are two types: parallel beta pleated sheets and anti-parallel beta pleated sheet with respect to N and C terminus. So it is like this, and C terminal, they are known as parallel beta pleated sheet. And if it is like it is known as anti-parallel beta pleated sheet. They are stabilized by the hydrogen bonding between carbonyl oxygen and amide nitrogen. They are stabilized by interchain hydrogen bonds, while in alpha helix they are stabilized by intrachain hydrogen bonds. So in beta pleated sheets, it is of two types, parallel and anti-parallel. These are the extended sheets. It is named beta because it is second in a discovery by Pauling and Corey, which gives the structural classification, that is Pauling and Corey. They give, it is second in the discovery. This is the first in discovery, that's why alpha helix. It is second in discovery, that's why it is known as beta pleated sheets. Okay. Example of beta pleated sheet is carbonic anhydrase. Carbonic anhydrase. It contains both parallel and anti-parallel sheets. Then silk fibroin. These are the example of beta pleated sheet. The turns, bends and loops, these are mainly involved in joining of these two secondary units, two secondary structure. They are involved in the joining of these two secondary structure. These are the short chain peptides, turn, bends and loops. These are the peptides which are involved in the joining of two secondary structure. There are some super secondary structure also which are involved in with the DNA. They are known as zinc finger motif, then leucine zipper motif and helix turn helix motif. These are known as super secondary structures. Okay, so this is about the beta pleated sheet and secondary structure. They are stabilized by hydrogen bonds. Most important is the hydrogen bond followed by hydrophobic interactions, electrostatic forces and van der Waals forces. Now coming to the tertiary structure. Tertiary structure, it is a functional unit. Functional unit. It is formed by folding of secondary structure to form a 3D orientation. Folding of secondary structure. Example is, most of the enzymes, they are tertiary structures. Enzymes. It includes alcohol dehydrogenase, lactate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, malate dehydrogenase. These are the examples of tertiary structure. Also myoglobin. It is a tertiary structure of protein. Okay. Primary, secondary and tertiary structure, they involve single polypeptide chain. While in quaternary structure, when two or more than two polypeptide chains are kept together, it is known as quaternary structure. Example is hemoglobin. Hemoglobin is an example of quaternary structure. Okay, hemoglobin, it is composed of 2 alpha chains and 2 beta chains. Quaternary structure of protein. These tertiary and quaternary structures of protein, they are stabilized by hydrophobic interactions, then hydrogen bonds, electrostatic forces and van der Waals forces. So this is about quaternary structure, and this is all about primary, secondary, tertiary and quaternary structure of protein. This is an important topic in biochemistry, and this is sufficient for your university examination. Keep watching. Thank you.